Start.
Good afternoon, everyone. It gives me immense pleasure to extend to you a very warm welcome on behalf of Department of Zoology and Internal Quality Assurance Cell of Gurudas College for today's webinar on Physics of Life Science, a Perspective. First of all, I welcome our respected Principal Madam, Dr. Mousumi Chatterjee, our IQAC coordinator, Dr. Triparna Mojumdar, honorable guest speaker. Today is our honorable guest speaker, Mr. Bidesh Chatterjee, who is our ex-student also, all the faculty members, all the delegates, and last but not the least, our students. Now I request our principal madam to say few words.
Good afternoon, everyone. Today's speaker, Mr. Bidesh Chatterjee, all the delegates, our faculty members of Gurudas College and my dear students. It gives me a warm welcome for everyone of you present in this webinar in the interest of our college committee. This is our fifth webinar in this period, that is rather during this challenging period. It is an endeavor which is worth appreciating. Today's topic is physics in life science or perspective. Actually, physical science, that is physics also, concerned with the study of natural but inanimate objects. These sciences include astronomy, physics, chemistry, and sometimes earth sciences. Life science, on the other hand, is the scientific study of living organisms, including animals, plants, and microorganisms. At first glance, it may seem like physics and life science are polar opposites. How can there be any overlap between the sciences of living organism and inanimate objects? But the truth is a little more complex than that. For one thing, the goal of physics is to explain everything in the universe on fundamental level. In theory, if physics ever succeeds at creating a so-called theory of everything, this should not only explain why inanimate objects are the way they are. Living organisms are, after all, only made of chemical elements, that they are made of subatomic particles. That physics seeks to explain, but at least in the short term, the connection between physics and life science could be seen as weak. But weak does not mean that non-existent. There are several cases where biologists and physicists can find ways to work together. That field is called the biophysics, where physics, theories, and methods are used to study biological systems. Biophysics generally tries to explain a lot of same phenomena as biochemistry and molecular biology, but tries to do so numerically. Physics technologies such as X-ray crystallography, spectroscopy, scattering effect, electron microscope, and there are so many, can also be used in this field. Though these technologies are mainstays of physics research, but are less used in life science. Physics understanding of statistical mechanics and thermodynamics can also be used in biophysics to understand biological sciences. Anyway, I will not prolong my speech. Now I'd like to present our ex-students, Mr. Bidesh Chatterjee, PhD scholar of Okinawa Institute of Science and Technology, Graduate University, Japan. He is going to deliver his lecture on physics in life science perspective. His talk today is a reflection on the outstanding work he is currently involved in. Thank you, Vinesh, for taking time out for bringing us the latest advances in the field of biology. I sincerely hope this webinar is going to be very fruitful and enjoyable for all of you, and especially for the students of biology. I offer my good wishes for the stakeholders. Now over to the organizer for proceeding the webinar. Thank you.
Thank you, madam, for your valuable words. Now I request Dr. Mojumdar, our ICAC coordinator, to say a few words.
Good afternoon. On behalf of Internal Quality Assurance Cell, Gurudas College, I welcome you all to today's webinar titled Physics of Life Sciences, a Perspective, jointly organized by the Department of Zoology and Internal Quality Assurance Cell, Gurudas College. During the pandemic, social distancing has become the order of the day. To continue with our academic pursuits, we have been organizing webinars. This session is the fifth webinar organized by us. I thank our librarian, Dr. Joydeep Chondo, and his team for giving us technical support in organizing these webinars. Today's webinar is a bit special for us since the speaker, Vivek Strategy, has graduated from the Department of Zoology of our college. Presently, he is pursuing his PhD from a reputed university in Japan. My best wishes are with him. Without further delay, I request to continue with the discussion. Thank you.
Thank you, Triparnadi. Now I invite Dr. Babli Haldar, HOD of Zoology Department, to introduce our today's speaker. Over to you.
Thank you, Dr. Polomi Devbhumi. Hello, everyone. It's a great pleasure for me to introduce our speaker today, who is going to talk to us about physics of life sciences, a perspective. Today's speaker is a remarkable contributor in the field of biophysics and developmental neurobiology. He was a university topper during his bachelor in zoology in the year of 2013. Sorry. Yeah. Due to some technical glitch. Uh, he received Shoroshi Bala Chaudhuri Memorial Scholarship in the university in the year of 2014, and he was a university topper during his bachelor's in zoology in the year of 2013, which he did from the Gurudas College. In the subsequent year, he received Biotech Rice Scholarship from West Bengal Biotechnology Development Corporation. He completed his MSc from University of Calcutta in 2015. In 2016, he received the Chopok Master Scholarship from Embassy of France in Italy. He had done internship from ISA Kolkata, and then he did his master thesis from University Pierre A. Marie France. He was a rotation student in membrane cooperativity unit and nucleic acid chemistry and engineering. Am I audible?
Yes, yes.
Am I audible? Yeah. He was a rotation student in membrane cooperativity unit and nucleic acid chemistry and engineering unit at Okinawa Institute of Science and Technology, Japan. He has presented his research finding in various conferences at Italy and France. Currently, he is pursuing his PhD research at Okinawa Institute of Science and Technology, Japan, where he is studying the process of lens morphogenesis in zebrafish. I think some technical ways. Am I audible now? Ravindra?
Yes, yes. Please continue.
Yeah. His thesis is about understanding the role of mechanical forces in regulating epithelial proliferation. He also studies the role of YAP as mechanosensor gene in the regulatory process. He is examining how YAP expression is lens epithelium is regulated by the mechanical tension of the epithelium. Let me welcome our guest speaker of today, Mr. Bidesh Chatterjee. Now over to Mr. Chatterjee to give us a glimpse of his research. Thank you.
So, do you see my screen? Am I live?
Yes.
Okay. So, thank you so much for those kind words to all the organizers. I would like to take this opportunity to thank them. And I'm really grateful to them for giving me this opportunity and present my work. Well, my name is Bidesh Chatterjee, and at present I'm doing my PhD at the Okinawa Institute of Science and Technology. This university is a tiny little, it's situated in a tiny little island in Japan. And here I'm working on physics for life sciences. This is something that I want to talk about that I have learned in the last few years. I joined my PhD in the year 2017, and before that, I was introduced to this topic where we were just trying to understand how we can approach problems and questions in biological sciences, and with the perspective of physics. And today I would like to share with the students what I have learned so far. Well, the intention of this seminar is basically to familiarize the students of classical biology, like zoology and botany and physiology, and even biochemistry. This perspective, which at least for instance, I missed it. And this is something that has been a learning experience for me, and I'm still on a learning curve. So, without further ado, let me start my presentation. And one more admission, though, that is, this is the first time ever I'm doing a webinar. It's a bit strange. I think it will grow on to me with time. So, let me go on to the first thing that I would like to show you is basically a video. As we see the video, we will see, and then we can talk about it, what's really going on this video, on this clip, and see how this presentation goes. Okay, so before I proceed further, I just want to make sure that the video was audible and visible to the audience. Jodip, sir, if you could just confirm that the video played and it was audible.
Yes, yes, it is audible.
Okay,
It is
thank you so
audible.
much. Okay. Okay. So let's now we are going to again. I mean, I do. So what you saw is that it's actually quite a dramatic representation of cells moving basically in a zebrafish embryo. So this clip was produced at the EMBL. Where these every dot is basically representing a cell with their different speeds. And what you see is this these cells in the embryo, they start not only dividing and becoming more and more in number, and but they start a very specialized movement around the embryo. And as they move, what is happening is that these cells, they are rearranging themselves into the three germinal layers, which is later going to form the different parts of the body. And it's actually less than half a day, almost like by 12 hours. What you see that the cells have rearranged themselves. They have established an anterior and posterior polarity in the animal. So there is an axis that is to be formed. The polarity is there, and you can already see that there are certain parts like the head and the eye cup. They're almost visible. Let me see if I can get the laser pointer. Yeah. So what I'm trying to convey to you is that during development or in life in general, cells are extremely capable entities. You know, they can go through so many reformations and to give rise to this variety of other cell types. And think of this embryo, for instance, this all started with it was just one cell. And then through the process of development, what happens is more cells are added to this entity, and not only that, each time a cell is added, those cells they have a specialized function, and these specialized functions are served by the different shapes and size and forms that they have. And this is in a multicellular organism, though. This pattern is visible throughout the animal or the living entity that we have here. Let's, for instance, here are the prokaryotic cells. So look at the versatility and the range of shapes and sizes present. Next, this one, it's a beautiful artistic representation of the range in size and shape of protists. For the comparison, what you have here is the head of a nail, and as a scale here, and you can see the various types of protists that are there. Maybe we can even identify a few. I think this one is Paramecium. And not only in single cellular organisms, when we come to multicellular organisms, look at the variety of cells that we have in shapes and sizes. So these are starting from here, this is a red blood cell, this is probably a fibroblast, this is a neuron. And so all these different cells in a multicellular organism, they all have different shapes and sizes, and it's really needed for them to do their function. For example, the red blood cell actually it's a very interesting cell body. What happens is, as we know, there is no nuclei in there, and it's almost like a ghost membrane. And what allows it, what is allowing the cell, what I mean, the structure of the cell, what happens is because of the structure, the cell itself can go through enormous degree of shape changes. You know, they can go through those tiny little pores in the vessels and enter into the tissue and do whatever they have to do. Same for the other cells. Okay, let's. I have a little tiny little quiz, not a quiz. I don't take it so seriously. Can we identify what cell this is? Students of zoology. I think this is a podocyte that you find in nephron. And how about this one? This cell is basically a rod cell here. But again, what I'm trying to really tell is that cells in multicellular organisms they have enormous versatility, and in terms of their shapes and sizes and their function. This adds to their function. Without this striation, the rod will not be able to do what it needs to do. So where is this versatility in cells and in a broader term in tissues come from? So for this, I'm going to take a case study, a very classical one that we always study in developmental biology. is the formation of neural tube. So let's look at this tissue formation. What's really happening? So in the first stage here, you have the neural plate, which has been specified. It's a part of the neural ectoderm. And what's going to happen is this these bunch of yellow cells, through a process of induction and cell-cell communication, they are going to form a neural groove in the next stage. And then what happens is this neural groove is going to invaginate and it's going to form a neural tube and it's going to pinch off from its parent neural plate. But in the process, what really happened? What happened is this flat sheet of cell here, which was a neural plate, is now transformed into this tubular shape. Now, how did this happen? Well, we have various mechanisms and various explanations for this, and those machineries are very much at work. So let's look at the checklist that we have here. The first thing you would say is differential gene expression, which is what? That how do these cells in the neural plate know that they have to become the neural tube? So there has to be these differential genes need to be expressed at different cells that lets them know what is their fate regarding their position in the embryo and the time of gene expression. This is very important. And another classic phenomenon in developmental biology is that of an induction and competence. So a responder will react to an inducer through cell-cell communication, and through these cell-cell communication processes, what happens is they will commit to a cell lineage. And after this commitment has been made, it will be specified, and then finally the fate is brought about. But with all these different machineries and explanation, can we really my the first question that one asks is it really with all these machineries that we have available for a cell or for example a tissue or a part of a tissue, how do you even try to explain the formation of a tube from this flat sheet? So let's take a simpler experiment that we can do that all of you can do with me. So this flat sheet of paper is basically your neural plate. Can you? I hope so. Is it? Am I visible, sir? Sorry, sir.
Yes, yes, yes. Please continue.
So,
Yes, please continue.
so I have this flat sheet of paper with me, and if I imagine this to be the neural plate, what's happening is this flat sheet of paper is folding, and then it's forming this tubular structure, which is going to be the neural tube. Now, in order to do this, what I did, I actually applied forces along the two edges, and this external mechanical force is allowing the flat sheet of paper to form into this beautiful tube that will later form the central nervous system, I mean, empirically. So, this is exactly what's happening here. You have the neural plate, which was a flat sheet of tissue, and through some way there has to be mechanical force that is being applied or being produced at these two edges of the sheet that allows it to form this neural tube. What I'm trying to say is these changes that are these machinery, the genetic and biochemical machinery that we have at work during development is not sufficient to bring about morphological changes. You need external mechanical force whenever there is a change of shape. And where is this mechanical force coming from? For instance, the piece for this piece of paper, the force was coming externally because it's me who's applying the force. The paper is not doing anything. I am applying the force externally so that it falls upon itself. But in case of tissues, what happens is the cells in a tissue, they are very much capable of not only producing force themselves, but they also respond to mechanical force. And this response to mechanical force is not only there just a matter of fact to change the shape of the cell, but in response to mechanical force, a cell can also change its gene expression pattern. To give you a bit clarity on this, let me go on to my next slide. So here it's the classic signal transduction pathway. What you're seeing it here is basically a protein called YAP, and it has another similar homolog called TAS. So what happens is that these YAP and TAS proteins, These are transcription factors, and what they do is they can go in and out of the nucleus. When the upstream pathway or YAPTAZ pathway is active, what happens is YAP is phosphorylated, and as YAP is phosphorylated, it is degraded, so it cannot move into the nucleus. When the upstream pathway is not active, YAP moves into nucleus, and when it moves into the nucleus, it binds to its receptor TEG, and they bind to the DNA, and specific gene expressions are brought about. But what's interesting about this slide is that the upstream, what is really the ligand here, we don't know what is on the upstream or the most upstream sequence of yaptas that is making the this cascade to go down, and then the yap moves in the nucleus. One good study that showed that yap or its expression is very much dependent on the the mechanical stimulus that it receives from the system. What they did is they cultured cell, and then they played around with the extracellular matrix of the culture system. So what they did, they were playing with the matrix stiffness. When the cells were grown in very stiff matrix, what happened is the yap moved into the nucleus. So when you have the cells growing on in the culture system where the matrix is very stiff, it's very hard substrate that the cells are going on, the yap moves into the nucleus. You see the red particles, they move into the nucleus, and these cells will form bone cells. But at the same time, when it's a soft matrix, the rigidity of the substrate is very low. The cells form fat cells. You see the house. It's remarkable that just by playing with the rigidity of the substrate on which cells are growing, you can change the fate of the cell without even touching an inch in the biochemistry of the cell. So what's really happening? How is this mechanical force that is transferred as the matrix rigidity is being translated into something like gene expression? So this is exactly where physics and biology come together, and this is something that we call mechanotransduction. That is the transduction of mechanical force into genetic pathways that bring about biological responses. So, to give you another interesting example, here what they were trying to see if mechanical stretch can really affect a formation of tumors or the lack thereof. So, what they did is they take a mice and they just stretch the mice every day for 10 minutes. So the mice actually had skin tumors and they used stretching as a therapy. And after stretching the mice only for 10 minutes every day, they saw a significant change in the tumor volume. You see, so it's almost like yoga for cancer. And as you keep on doing this stretch therapy, the tumor volume it actually gets better and better, and it's not as without stretch you can see how the tumor growth is growing up, and with stretch the tumor volume is not as significant, it's significantly lower than without stretch. So this is a very direct example that shows you how we can use the mechanical response, the mechanical stimuli, even as a therapeutic element in our medical practices. Now, if you have any question, I would like to. If you have any questions so far, I hope I'm not going very fast. If you have questions, please let me know in the live chat. I have ways to answer your questions, hopefully, because it's very difficult for me to tell without seeing the audience if I'm going too far. I'm trying to not go very fast. So please, if you guys have questions, let me know at any time. So let's move on to the next slide. Is what are these mechanical forces in biology? How do we really probe mechanical force in biology? What's going on? So let's think of two components that allows us to look at mechanical force being applied to biological phenomenon. So let's take a look at this sheet of cells, sheet of tissue, and it is almost like an epithelium. And what if you apply mechanical force to it? So you're applying force. What changes does it happen regarding its area? You can apply mechanical tension that allows the tissue to stretch in area, or you can compress it. Again, it's a change in its area. Or what you can do is you can shear it. It's almost like putting the tissue between two plates, and you move the plates in two different sides, and that leads to this shear stress. So these are just in a few examples of the different kind of mechanical tension or mechanical stimulus you can put into a biological system. And then we can study how these different cells or the tissue in general, in totality, responds to the mechanical forces applied to them. Let's take a case study that the system I'm studying right now is the zebrafish lens. So the zebrafish lens, it's a very interesting. I mean, it's not very interesting. It's a very simple structure. It's spherical in nature. See here, you see in the zebrafish eye. If you do a section, you have the beautiful retina around it, and then this is the lens here, right here, the spherical, clear organ, and then you look at the organ how it looks like. The lens is of course spherical, in cross section it's circular, but look at the anatomy of the lens. So it anteriorly it has this covering of a monolayer of epithelium in orange, and posteriorly it is covered by these green fiber cells. So the fiber cells make up the entire anterior of the lens fiber core. Even here there are fiber cells. Another feature of the lens is that in the lens, only the epithelial cells, that is the orange cells, they can proliferate. And as these cells proliferate, what happens is they will pass the equator. And as they pass the equator, they start to differentiate into these long fiber cells that form the lens core. And this is almost like an epithelial to mesenchymal transition. And the epithelial to lens fiber transformation is dependent on so many important and almost all those major uh, cell communication processes, the cell signaling pathways. And it's very complicated to understand exactly what is it that regulates that plays a key feature in epithelial to fiber cell transformation. One very good component is the fibroblast growth factor that our lab studies. But for me, more interesting part is this lens epithelium itself. So let's take a look at the lens epithelium the way we did for the neural tube formation. So this is how the lens epithelium looks like. Again, it's starting off from a flat sheet of cells, the lens placode, and due to the developmental processes, what happens is this lens placode is forming. It's not invaginating that you saw in the neural tube, right? What's happening is these cells, they start first thickening, and as they thicken, they just delaminate from the cells, and now it's a spherical structure. You see, here the formation of from a flat sheet of cells to a spherical structure is not invagination that we saw in neural tube formation, but it's more of a delamination process. So the entire cell just kind of, so here is this almost like a vestibular cell that's making this these two different tissues conjoint, and then it just separates, and then this lens itself is going to grow on its own, and as it grows it forms this beautiful structure where the epithelial structure, the epithelial monolayer in anteriorly is already established. Here you can see in green, and the lens fiber cells are here in blue. Okay, so my here I'm trying to understand what is it that is regulating the lens epithelial proliferation here, because as we saw earlier that only the lens epithelium that can proliferate, and as they proliferate they transfer and pass the equator here. So you have the lens equator, and the lens epithelium is proliferating. As it proliferates, it will pass the equator and it differentiates into green lens fiber cells. But the lens fiber cells cannot proliferate at all. So that means that in order to keep the spherical shape intact, there must be a regulation of cell proliferation around the lens contour that will regulate the rate at which the lens epithelial cells not only divide but also pass the equator. And this is very much important for the entity or the integrity of the organ. Imagine if there is a localized hyperproliferation of the lens epithelial cells. What will happen is that the epithelial will just it will extrude out on its own, and the spherical nature will not be there. And the spherical nature is very much important for fish because they are aquatic creatures. So let's take a closer look at the tissue level. What's going on in the lens epithelial sheet? So epithelial sheet at first is just a flat piece, like as we saw in the neural plate, it was a flat sheet of cells. And then through development, what's going on? So instead of forming a tube where you just provide mechanical force on the two edges and they will fold on it on their own, the epithelial is forming a dome-like structure. Okay, so this is a very different kind of morphogenesis, and here the mechanical force its arise and its distribution is not as simple as we saw in the neural epithelium. So this is exactly what we are trying to understand in my lab, or at least in my PhD. How do you really kind of a how do you explain the formation of a epithelial dome from an epithelial sheet? What is the role of the mechanical forces here, and how these mechanical forces arise, and how can we quantify them? So the question is, are there ways to quantify mechanical force in a developing system? So I use two different tools that can be used to measure or even quantify, or at least qualitatively, to look at the mechanical force that is arising at developing tissue. One is called laser ablation, the first one example that I will talk about. So, what is laser ablation? I will show you a little clip, and what actually two clips. The first clip here, it's a single cell where laser ablation is taking place, and just see what happens. I'm going to play this again for you. So, there. So what we did is just we applied a very high powered laser at a single point, and that high power laser, what it did is just burnt this part of the cell where the laser was focused in 3D. So let's take a look in instead of this, what happens when you do this in a sheet of epithelial cells. So this one is a bit quick, so I'm going to play this a few times and see what happens. You just make a cut on the epithelium there. Once again, there we go. So what's going on here? So what we are doing is, instead of pointing at a single point in the cell-cell contacts, you draw a line, and across the line you shine that very high-powered laser that cuts the cell-cell contacts between them. You know, and as these cell-cell contacts are cut, you can quantify how much mechanical force that was there before you cut the cell into different pieces. So here, let's look at the principle of laser ablation. So this is my epithelium, any epithelium. And if I look at this trijunction of cell-cell contacts right here, how does it look like? So we are taking these three cells in isolation, and now we are thinking about a single point, any apex point, and what is the force distribution there. Now, if the epithelium is static, that means there is no net movement of this point, that the cell is not very elastic, or this point is not very movable. Then the force distribution is going along the three axes is not going to be proportional, but at equilibrium, the summation of force is going to be zero, since there is no movement there, right? And now when you cut this into a when you by laser ablation, so here this is, think about this. So I have the trijunction here, and at this point I'm going to shine my laser. So this is the epithelium. At this red arrow, the laser is being shown. As you shine the laser here, what happens is there's a cut. So there's a discontinuity of the cell-cell contacts, and then what's going to happen, because of the elasticity of the tissue on its own, these two yellow points, they're going to go further and further apart. So it's basically almost like a tug of war. The cell-cell contacts are the tug of war, and then there's a lot of force in there, and then with laser you just burn this point and it goes away. You see, this is what we are looking at here. Basically, this is what's happening. There's a tug of war going on at the cell-cell contacts, And with a very high focus laser, you just burn a part of this, and this react. And what happens? The neighboring cells are going to react. So here here it's not a video, but what I'm trying to convey is that after burning, that you can see that these two yellow points, they're going further and further apart. Now this tells you what? This tells you that the speed at which the cell contacts go apart after a laser ablation. This is a direct reflection of the mechanical force or the mechanical tension that was present before the laser ablation. Once again, so imagine if you are at a tug of war competition, and what you're doing is you're playing this tug of war. The more force you apply to this, the more and more force I apply to, then I now I bring a scissor and cut this tug of war. The more force it was there before I cut the tug. What will happen is the faster you will go apart. It was just a bit floppy. There would be not much movement around the tug of war or the cell contact. There would not be a lot of movement. After you do the laser ablation, the higher the tension was before the laser ablation, the faster the 2 points are going to go apart. So this is not exactly as direct measurement of the mechanical tension of the epithelium, but it's an estimation of what is the respective mechanical tension in the epithelium qualitatively, right? So that means after you do the laser ablation here, you will see the vertex displacement, and you see, why do you think there is a plateau here? So as you, with time, what happens is these 2 points are going to go apart, and what as they go apart, they're going to form a plateau. It's because it's in a low Reynolds number environment. That means there is no time there. So after a point, it just stops and it reaches the plateau. Anyway, that is not very important. What I am trying to say here is laser ablation gives you a qualitative measurement of the mechanical tension or the profile of the mechanical tension in an epithelial sheet. Using this, you can design different kinds of experiments that will allow you to measure the mechanical tension present in the epithelium sheet. Right. This is what I do in my PhD, and I'm trying to develop methods that will allow you not only if so. What I can do with this is basically, let's go back to the structure of the lens. The lens is a spherical structure, right? Now, if I want to measure if there is an heterogeneity of mechanical force, what I can do is I can make me laser ablation at different orientations and then measure the retraction rate or the displacement velocity. Depending on higher and lower mechanical tension regimes, the displacement profile or the velocity profile at different regions will be different, and that will give you a map of mechanical tension on the epithelial on the lens epithelium. Imagine this is my lens epithelium. This is a dome-shaped structure, and this is the pole of the lens, and this is the equatorial region. One can always assume that maybe the mechanical tension along this contour at the equatorial region is much greater than the one here at the pole. And by designing the laser ablation, you can actually measure that, where there is a more tension and where there is a profile of less tension. That allows you to map the entire epithelium in terms of mechanical tension. So this is what I wanted to talk. This was one of the first tools that I use to quantify mechanical tension in epithelium using laser ablation. You can do it at single cell or even multiple cells, like the researcher has done here. And I'm trying to employ this in lens epithelium, and hopefully we are getting very interesting results. Um, the other part is I use as a FRET-based sensor. So this is a modification of fluorescence energy. So FRET stands for Foster Resonance Energy Transfer, right? So what happens here, the principle is very much the same as fluorescence excitation and emission. So you have a photoexcitatory molecule, or in this case, a donor. When you apply a particular wavelength to this donor, it goes into a higher energy state, and after that it will come back to a low energy state. After, when it comes back, so this is the excitation energy. When you with this excitation wavelength, when you make it reach this higher energy state, and then it's going to travel back to its ground state, the S0 state is going to release another wavelength, and that is what you see as a basis of your fluorescence. Now imagine this emission that you get from this donor can be taken up by another molecule. So here we have an absorption for CFP. It absorbs a certain wavelength, and then it will emit a certain wavelength. If the CFP and the other molecule, they come very close to each other, the emission of the first donor can be picked up by the acceptor, and then this phenomenon is called FRET. So instead of emission of the cyan light, you will see emission of yellow light. That means whenever the donor and the acceptor are very close to each other, the fret occurs, the phenomenon of fret occurs. And using this principle, we can try to understand if there is any intramolecular tension profile that is present across lens epithelium. Let me give you the example. So what you do is you have this E-cadherin molecule, right? But you've engineered it and you have put a donor and an acceptor in between. So you have the E-cadherin molecule. Here is the cytoskeleton where the intercellular part of the E-cadherin molecule is going to contact, and the extracellular part is out there out of the membrane. This is the transmembrane region, and you have the extracellular domain which contacts the other E-cadherin molecules. And in between, you have a donor and an acceptor. What's going to happen is whenever there is a high mechanical force, and one more thing of the engineering is that you have the donor and the acceptor, they are both joined by this spring-like protein. This is taken from the spider silk protein. What happens is whenever you apply force, the spring here, it extends. So this is what's going to happen. Whenever you apply force to the E-cadherin molecule, either externally or internally, the elastic linker is going to stretch. You see this transformation? That tells you what? If my donor and acceptor, when they are very close by, I'm going to see fret. So because since they are very close, the donor is going to be excited by a certain wavelength, and because of the phenomenon of FRET, the acceptor is going to take it's going to take the emission wavelength, and it will give its own emission profile. But when it's a high mechanical tension there that is being given on this extracellular on the E-cadherin molecules, this is what happens. You will see no FRET. So by changing this, by using of the very principle of FRET, which is an optical tool, you can actually measure the intracellular tension profile of in the cell or the epithelium. So these are the two tools that I want to talk to you about regarding that allows you to measure mechanical forces in epithelium. And with this, the summary of this, that mechanobiology, it's an emerging state of study that allows you to study the interplay of biochemistry and mechanics, especially in developmental biology. It is exceedingly well established that without mechanical force, it's you need mechanical force in order to understand and explain developmental processes. And it's an interdisciplinary approach. And of course, to do this, biologists need to be in contact and in dialogue with scientists from physics and mathematics. A lot of computer science is needed in order if you want to model something. And it's a rapidly developing field, and it's the experimental tools are they're extremely modern, and they're high tech, and they're state of the art tools that are there in the field. So what I'm trying to tell students that please look into this. You might find this interesting, and then you might want to delve deep into this. It's a very interesting field, and it's an emerging field. There's a lot of scope for everybody. So this is my very short presentation for the students. With this, I would like to really thank everybody who has made this happen. Principal Ma'am, Dr. Moshmi Chatterjee, the IQAC coordinator, Dr. Triporna Muzumdar. Thank you so much for this arrangement. Dr. Joydeep Chandu, thank you so much for helping me at every step of this presentation. And my teachers, Dr. Monica Mehta, Dr. Babli Haldar, and Dr. Poulomi Bhumik, they have been an enormous help for me to present to you this webinar. So with this, I would like to take um, questions that students might have. I understand I might have gone a bit too fast. So if you haven't understood anything, please let me know in the live chat, and I'm more than happy to answer for you. Thank you.
I would like to thank Mr. Viresh Chatterjee for such an amazing and enlightening lecture. Although I am not from the field of biophysics, but the lucid presentation made it so easy to understand every bit of it. In his study, he wanted to dissect out the role of the physical and mechanical forces that govern the basic biological process. The presentation again showed how the inherent linkage of biology and physics are helping the modern scientific advance. The main beneficiaries of this lecture are our students. They could relate with their subjects taught in the classroom and the content of the presentation. This exposure will create an interest for biophysics in the young minds, and we can expect many more promising researchers in the days to come. Thanks, everybody.
Okay. So there's a question if I have tried mechanical stretching experiment on tumor growth and other animals. So I don't work with tumors. Our lab doesn't work with tumor growth. It's a developmental neurobiology lab. But tumor growth, it has been shown even in tissue culture you can do that. Um, but I haven't personally done it. It's not something that I work on. What is the mechanical force associated with autoimmune diseases? That's a very interesting question. Autoimmune disease. So mechanical forces. Think about what is the most important. I don't know whose is this from a research perspective or just a general query, but think about the autoimmune disease. What are the cells that are present in the immune system? The immune system cells are extremely motile. So these motile cells, like there are so many different kinds of the immune cells. For them to, in order to go through and pass through the different crevices and then capture a pathogen, there needs to be an immense interplay and dynamics of the cytoskeletal system. So without the cytoskeletal system, of course, I should have made this a bit more clearer. The source of mechanical force in cells, it is the cytoskeleton. Without the cytoskeletal system and without the motor proteins, there cannot be any source. What are the motor proteins? The motor proteins are myosin and the kinesin and dynein. These are the sources that give you the mechanical forces. When you contract a cell, not only muscle cells, but in non-muscle myosin as well. And you have the actin polymerization. It's a source of great strength. Imagine a cell where you have these chromatids and they need to be pulled apart from the center to the periphery when cell division happens. So the amount of uh, power that you need, these are not possible without the motor proteins. So these cytoskeletal elements are very much important in wherever you want to produce or respond to forces. How can I explain mechanical force not found in? How can you explain mechanical force operating in? So the mechanical forces can be it's quite easy. You don't need to have a malignant cell culture. Imagine you are playing with the showed in my as I showed in my the experiment with the app and Taz, where they were just trying to understand it. They were just normal cells. They were not malignant cell cultures. They are primary cell cultures or even stem cells. You just grow them on different on the of matrix of different substrate rigidity. And this rigidity is actually what? It's a source of mechanical force. You can also play with mechanical force in cell cultures by reducing the size of where it's growing. So if you don't have, for example, you can constrict a cell. You give it exactly a very little tiny surface area where it can grow. That is also a mechanical force. That's also a source of mechanical force to the cell itself, and that is important in determining the fate and its function. What else? Okay. So here's a question: stretching and exercise are proved to reduce tumor growth, aging effects. But what is more effective here, the physical change or the physiological changes related to metabolism? That's a very, I think it's a bit difficult to answer at this point. What is more important? What I'm trying to say is that uh, um, it's not a competitive force. For example, I won't say that it's possible to turn a cell into a tumor cell just by applying a mechanical force to it. But the it's not exactly irreversible. So it's a constant interplay between mechanical forces and the cellular machinery. But I don't want to make any claims at this point. Okay, AFM is. I really want to talk about AFM. This is very interesting because what people have done is they wanted to understand. I really like the experiment. So they wanted to understand what is the substrate rigidity of brain. So you have different neurons in different parts of the brain, and for example, think of the optic chiasma. The one neuron is crossing one end, and the other one is coming from one end to the other, and they form a perfect chiasma. So how does the cell know, or how does the neuron really find its path? What is guiding it? So they thought maybe it's the substrate rigidities of the brain that is very much important for guiding the neuron. So what they did is they would expose the brain of the frog or the amphibian, and they did atomic force microscopy. So they just tried to find out the map of the brain where it's more rigid and where it's less rigid, and they found that actually there's a profile of substrate rigidity along the surface of the brain, and that is almost guiding the neurons to find their path. Which stimulates the more cells during development and stimulates during development and expansion ahead. So, so far, at least in my animal models, during the primary stages of development, I don't know of any cilia and flagella. I think cilia and flagella come much later, at later stages, which are important. For example, if you want to create a flow of fluid, there it's very important to have cilia right there, and cilia is very much important for sensory purposes. A flagellated cell, I am not very sure of, but if you think of just cilia and flagella there, it's also something that refers to the low Reynolds number where the cells are, and the physics is there is very important. I'm just going through the all the messages. If there is a question that I have missed. What? Okay, how intercellular mechanical forces help us know about the external force, which is helping in changing the shape of the tissue. How intercellular mechanical forces help us to know about the mechanical force, which is Helping in changing. I think probably you're asking about if uh, there are studies where mechanical forces have been shown to change the shape. For example, I'll give you this instance. Um, I don't have the slides with me, but it's the optical. Actually, it was my master's thesis where I was studying the olfactory system of zebrafish. And then what you see there is it's a phenomenal. It's a okay. Maybe I can play a video, and then I might have the video with me. Let me see. Okay, well, I don't have the video with me. Um, But what happens is there, so in the olfactory placode of or zebrafish, you see all these cells, they form a bulbous structure. So imagine this is the head of the fish, and the cells first migrate, the optical placode cells, they will migrate and form a bulbous structure. And one very interesting thing that happens is that the cells, so in a neuron, what happens in a neuron, you have the cell body, and from the cell body the axon comes out, and the axon tries to go through, and it's going to find and form a synapses. In olfactory placode of zebrafish, it's very interesting. You have the wall of the brain, and you have the bulbous structure of the olfactory cells. And then what's happening is, instead of the axon moving out, what happens is the head of the cell body of the neuron is moving away from the wall, from the brain wall. So I was trying to understand what is the role, what is it that's going on, because the researcher at first saw that there is no role of cell division, there is no role of cell death, cell growth, volume changes. None of these straightforward biological factors of a cell, they don't play a role in it. What was really going on? So then they started understanding maybe it's a mechanical force, that something is either pulling the cell apart from the brain wall, or something is pushing it so that it just keeps extending like a ball of dough, a flour dough. I don't know if it answers your question though, because but well, what I'm trying to say is that there are, of course, different instances where they have tried to understand biological and complete morphogenetic processes by way of mechanical forces.
I think there are no more questions.
Okay.
So, Professor Babli, please continue. Please unmute your mic first. Babli, please unmute your
Yes.
mic first. Okay.
Am I audible, Jodhipur? Am I
Yeah,
audible, Jodhipur?
yeah,
Okay,
yeah,
okay.
absolutely.
Thank you very much. Thank you very much, Dinesh, for your wonderful lecture and how you address it. Thank you very much for that. So let me proceed to the following phase. May
I
I
have
have your attention, please?
Case, ma'am, actually. There's a question from Case, ma'am. It says, what type of cytoskeleton is helping in morphogenesis? Uh, Normally, we think about the actin cytoskeleton at least in the first phase of morphogenesis. It's always the actomyosin cytoskeleton. The actomyosin cytoskeleton can it can work in forms of mesh, or there is also instances of shooting as in you would see a comet of actin cytoskeleton that just shoots off the cell, and you see these forces being present, and I mean, it can bring about a lot of dramatic change. But to answer your question, it's the actomyosin cytoskeleton that primarily works on. But there is also instance where microtubule, and both microtubule and actomyosin mesh of actin and microtubule are present at work. Yeah, I think I just answered it.
Okay, thank you, Videsh. There is an important announcement. To get the certificate, enrolled participants are requested to log in to our registration site, that is Gurudas College courseware. Click on today's topic from online webinars, where one can see the feedback form. After filling up the form, you can download the certificate directly from here. Thank you. Now, I would like to request Dr. Monica Mehta from the Department of Zoology to propose a vote of. Thank you.
and making it a success.
Bubbly, I think there is a problem of sound. I just restart it. Okay, please wait for some time.
Okay. Yeah, no problem. Please you can.
I would like
Before you start, Videsh, please stop your screen sharing.
Oh yeah, right. Thanks.
I would like to take this opportunity to thank Mr. Bidesh Chatterjee for a very informative, knowledgeable, and riveting session. I'd also like to extend my thanks to our principal ma'am, our IQAC coordinator, and our Teachers Council secretary. A very big thank you to our dear librarian who has given us invaluable support in setting up this webinar. Special thanks go out to all our fellow professors and colleagues who have made the time to attend this webinar. And last but not least, a very big thank you to all our students, present and past, who hopefully have gained knowledge about another aspect of life sciences. Thank you all for attending our webinar and making it a success.
Thank you, madam. With, yeah. Now I am audible.
Yeah.
Yes.
Thank you, madam. With this, we are concluding today's webinar. Thanks everybody for your participation and active engagement. Stay healthy and safe during the pandemic times. Thank you to all.
Thank you.